out of it.
We're just about there, a couple of minutes. Good luck. Good afternoon. So it's 3 o'clock, so I would suggest that we start. Welcome to this symposium on charting brain variability in health and disease using normative models. My name is Marquand. I'm an associate professor at the Donders Institute. On behalf of all the speakers, I'm very pleased to see you all here. I hope it will be a very fruitful discussion. We have four very complimentary talks for you today. I notice we have microphones in the audience, so please feel free to use those to ask questions, or put them in the app, and we'll try and have a hybrid question and answer session and see how that works. We'll take all the questions at the end. So if you have questions that are of a general nature, feel free to ask them. If they're for a specific talk, please indicate them as such in the app or when you get up to the microphone. So without further ado, let's begin. So could you bring up the slides? Thank you. So I'm going to give the first talk. My talk is entitled Shifting the Focus from the Group to the Individual. I would like to say straight away, I'd like to start with some acknowledgements. The work that I'm going to present today is really a team effort. We have a great team at the Donders Institute, and the credit for what I'm going to show should go to these people as much as to me, students, collaborators, and mentors, if not more. I'm going to talk about 3 topics today. So I'm going to talk about, give you some conceptual background. I'm going to talk about charting variation across the lifespan, and I'm going to talk about some kind of inferences that we can make on the basis of normative models. This is a very conceptual talk, and it will be followed up by some specific examples of the kinds of things that we can do with this modeling framework. So the classical starting point for normative modeling is conceptualizing it in a similar way to that we might think of pediatric growth charts, where we learn some kind of mapping between demographic variables like age and height, it keeps jumping, age and height or weight. So it's learning a mapping between some kind of non-biological variables in biology. So what we can do in a simple context is we can just translate these variables for things that are relevant to psychiatry or neurology or whatever disorder you're interested in, and they can really be any kind of measure you like. So we're not limited to just learning mappings between brain development, age and development and so on. We can learn mappings between symptoms, genetics, cognition, and so forth. If we do this in a fashion where we learn regional variation, learn a regionally varying model such that we have a separate mapping for each sample brain region, we can then build up a very rich space, a normative reference model that we can then use for subject-level inferences. And this is really the key innovation in normative modeling. We move from being able to say things about groups, group averages, to being able to say things about individuals. And that provides a very nice platform for subsequent stratification. Work from Sage Rutherford at the Donders is, she's really put in a lot of work to produce a comprehensive normative reference model for cortical thickness and brain volume in this case by aggregating data across 60,000 individuals. You can come and visit her poster tomorrow if you like. And the idea is that by aggregating data we can get good estimates of variation across the lifespan such that we can then translate that to new samples and make inferences in clinical populations. And it's quite important in this sense that the data, that the model is adapted on independent data, so that we can be sure that the model remains unbiased when we want to make inferences. Of course, we're not the only ones working in this space. There are other normative reference models for brain development that you might like to consider. In fact, this idea has been around for quite some time, and I'd like to just highlight this particular study. This is Gabriel Ziegler, and it was, we started working in this space maybe 2015 or so, but in this paper, really a lot of the core ideas were already in place in terms of learning variation across the lifespan, even getting down to the voxel level. So I'd just like to raise attention for this paper as well, and it's been somewhat overlooked by the field. So for the rest of the talk, I'm going to focus a little bit about, not so much about the estimation of normative modeling. There's lots of challenges associated with that in terms of modeling site variation. Johanna will talk about that in a minute, but also modeling non-Gaussianity, etc. I'm not going to talk so much about that. What I'm going to talk about is what kind of inferences that we can make using these models. So supposing we have a set of data points without any kind of knowledge, without fitting any kind of model to these data, we would be forced to make inferences with respect to the marginal distribution of these variables. So if we were to fit some kind of normative model, what we do is we effectively we're translating the raw data from raw values of cortical thickness, brain activity, etc., to population centiles. Now, why is that valuable in terms of in this particular context? It means that we reduce the variability so the normative model soaks up a lot of variation, and then we can perform, in this case, group level inferences with respect to these expected population centiles. Now, the outcome of that type of analysis is that we amplify group differences. So we have a purer signal that we can then use for classifying or detecting univariate group differences. And this is shown in a figure from Sage Rutherford, where you can see across all of the diagnoses, that's the columns, the magnitude of group differences is higher when we take these population centiles, the data that's been adjusted for population centiles. That's one thing that we can do with these kinds of models. Another thing that I think is a really valuable contribution is that normative models break the symmetry that are inherent in group comparisons. So if we were to have some kind of arbitrary population, and here I'm abstracting away from the details, what we do, whether we do it in a univariate context or using something like Fisher's linear discriminant, is we're effectively testing differences between group means. Now, in the presence of heterogeneity, you can see that the mean of the clinical group is actually not really a meaningful quantity. So we're trying to detect a mean that's not representative of any of the people within the population. And so by using this kind of normative modeling framework, we can translate from group level inferences towards individual inferences, and that provides us a way to get a handle on this kind of heterogeneity, the inter-individual variability that we see within these clinical cohorts. And if you want an example, there's a poster today. Amanda Worker has shown an example about predicting outcome in first episode psychosis at the level of the individual. And we've used this kind of formula in many different studies. This is work from Thomas Wolfers and Mariam Zabihi across many different disorders. And I'm showing you some examples so you can see on the top. In schizophrenia, on the left-hand side, we have classical group differences, a t-test if you like. And on the right, we have individual differences. So that's the proportion of overlap across individuals. And whether we consider the case of schizophrenia, where we have robust group level differences all the way down to autism where we have very few, in each case we see that the overlap across subjects is very, very low. And so this provides us a way to quantify individual differences at the level of differences at the level of the individual. Now you might say, well, this is a truism. We'll always see this kind of pattern, and I'd like to try to convince you that that's not the case. So for example, in dementia, where we have a much greater convergence, and we have up to 25% overlap between individuals in the medial temporal lobe, which is, of course, is one of the key regions implicated in dementia. So then the question is, so what can we do with that? We have these kinds of nicely calibrated Z statistics. I think they're particularly valuable if we estimate them at a very high spatial resolution. So we have a whole brain image that describes a deviation from an expected pattern, for example. So we can use these as biomarkers, and there's a number of different things that we could use these for. And this is again work from Mariam Zabihi, Charlotte Fraser, and Natalie Holtz. So the simplest thing is we could just train a clustering algorithm on the basis of these Z statistic images, and Mariam Zabihi showed that is a useful way to generate biotypes for autism. And in this case, one of the interesting things is you see that there are both increases and decreases in cortical thickness, which explains why we don't see many case control differences. We could also take a more sort of overlapping perspective rather than forcing each individual to belong to one and only one subtype. We might instead look for latent factors using some kind of matrix factorization technique, for example, ICA or some variant of factor analysis, and that would give us individual profiles for different participants that can overlap within different individuals. Another thing that we can do that is particularly interesting, and this is the topic of Charlotte Fraser's poster, is to use these as the basis for an extreme value statistics kind of approach. Now, extreme value statistics is an area of statistics that hasn't seen very much application in neuroimaging, but it's, I think, very appealing in the sense that we aim to quantify. I mean, the mean is not really informative about psychopathology. What we're interested in is we're interested in the most extreme values often. And you can talk to her about that at her poster. So this is a way to generate individualized risk scores from brain images. Another thing we can do with normative models is we can use them as a way to bind different kinds of data together. So supposing we have a particular normative model, for argument's sake, let's say we're interested in the left lateral ventricle. Now, that has units in terms of, expressed in terms of milliliters. And what we do in normative modeling, as I've explained, is we move from raw values to expected population centiles, this remapping step. And then suppose we were interested in combining that with a different kind of data. For argument's sake, let's say we're interested in combining it with cortical thickness. The units of these two measures are totally different. So it's arguable how meaningful it would be just to combine all these things into a single model. Whereas if we have everything in terms of expected population centiles, we have a really nice, well-calibrated model that, where all the features that we're including are on the same scale in terms of, plus or minus 2 being interesting in some sense. We can also use this in a longitudinal setting. So, for example, supposing we're interested in what happens over time, we might have data from subjects scanned at different time points. And the particular example that I'm going to talk about is for a longitudinal study where we have data from subjects that are actually acquired on a different scanner 10 years later. And what we're interested in doing is making inferences to connect these in a classical longitudinal framework. I'm sorry, the pointer keeps skipping. So it turns out that we can use normative modeling to play quite a nice trick in this respect. So we can, on the left-hand side, you can see in this figure, we have the raw data, and you can see quite clearly the site effect, whereas if we take it with respect to expected population centiles, we can make meaningful comparisons across scanners. And this is the topic of Pierre Berthier's poster. You can come and visit him to discuss the details of that. That's built on hierarchical Bayesian regression. These kinds of models also, another thing that might be of interest in many contexts is using these for actually detecting longitudinal change. And normative models become particularly sensitive in this context, and I'll explain why. So supposing we have some kind of normative model. And what I've been talking to you thus far has largely been about detecting samples that are extreme in some way, so a high centile or a low centile. If we have longitudinal data, what we'd like to do in many cases is to detect whether a subject is tracking its expected centile, his or her expected centile, or whether there's a centile crossing. In development, this would be termed as a failure to thrive. And the way we would, using a classical growth chart, if you like, you can make use of these things called thrive lines. So this is an actual growth chart for boys' weight. And these thrive lines are actually plastic overlays that we put on top of the normative growth chart. And the lines, I'm going to gloss over the details, but the lines you see are derived from the derivatives of the estimated centiles. And if we cross one of those things, then we would diagnose something like a failure to thrive. We can do the same sorts of things in normative modeling by accounting for the expected variation within our model. So we know we have some parameters. These are the centiles. They're estimated with some uncertainty. So what we'd like to say is that the difference is larger than the uncertainty in the model. And we do this and you can see this occurring here. We can do this in a Bayesian model, get nice different uncertainty, different types of uncertainty, separating modeling uncertainty from variation. So we can do this quite easily in a normative modeling framework. You can come and talk to Barbara Bokova, who's got a poster on this topic, and this one of the nice things about this approach is that she also takes into account test-retest variability. So that's another source of variation that we ought to take into account in this particular setting. The last topic that I'm going to introduce to you is again work from Mariam Zabihi. She uses, in fact, an autoencoder to get some kind of latent representation that we can then use as a basis for normative modeling. And one of the nice features about this approach is that it really takes into account multivariate structure. So in short, she estimates a normative model, an autoencoder on the basis of the Human Connectome Project data. As you know, that has good coverage of different brain regions, and then applies that in a transfer learning setting to UK Biobank, where they only have one of the tasks that is available in the Human Connectome Project. We do this, Mariam does this in a semi-supervised way, so that as well as in the loss function, we also account for age and sex, and that has the effect of biasing the latent representation that the autoencoder learns, such that the axes of variation are meaningful. And I'm showing here males and females are color-coded according to red and blue, and also we have the age of the participants encoded by the opacity of the different features. In this latent representation, we can then fit a flexible normative model, and that provides us the ability to get a multivariate estimate of change. And you can come and talk to Mariam at her poster to find out more details about that. So, take-home messages. I hope to have convinced you that normative modeling provides a platform for understanding individual variation. We can make use of big data cohorts to really map variation across the lifespan and across other types of variables as well. And shifting the focus from the group to the individual allows us to make many kinds of inferences which are very difficult to make in in other analytical frameworks. I'd just like to again acknowledge the people that contributed to this work. And here are the posters that we are, just to remind you. And I'd also like to draw your attention. We have all of the software tools that I've talked about just now are available online. We have, oh, my slides have gone. But you can come and check it all out online. And with that, I'll stop. Thank you very much for your attention. Okay, and so the next speaker will take, as I mentioned, we'll take questions at the end. The next speaker is Janine Beisterbosch. I'm very pleased to have her on board with this symposium. She's an assistant professor at WashU, and I'm very much looking forward to her talk, which will be a little bit more clinically oriented.
Hello, everyone. Thank you very much, Andre. And yeah, following on from that amazing introduction to normative modeling, I'd like to tell you a little bit about a project that's still ongoing in our lab that's using this approach to ask a more clinical question looking at heterogeneity in depression. So for this study, we are using data from the UK Biobank. Many of you have probably heard about it. It's a large population data set where the goal is to scan 100,000 people. And it's about halfway through, so already data from tens of thousands of people is available. It's what they call a prospective epidemiological data set, which means that people are not recruited into this data set with labels, so it's not like they're recruiting patients and healthy controls, but there's no specific inclusion or exclusion criteria, so it's a really population snapshot. And there's a lot of phenotypic and clinical data available. And so I'm really excited about the UK Biobank dataset, particularly from the perspective of being interested in mental health. There's been a lot of really valuable studies in our field looking at mental health and depression in particular. But when you've got funding to scan 100 people, you have to make some decisions about who you're going to scan. And usually it comes in the way of exclusion criteria for comorbidities or medication status or things like that. And what I think something like the UK Biobank offers us is that it's a real look at the population, which we know in the case of mental health is a lot more messy than a lot of those perfect studies. So we know that there's a lot of comorbidity, there's a lot of differences in symptoms. And so I'm excited to study real-world population mental health. So what I'm going to tell you soon is we started off thinking about symptoms. Then we went and took the benefits from the large sample size in multiple ways, including using the normative modeling approach. Then we quantified heterogeneity in a couple of different ways, and the thing that we're working on actively at the moment is to look at etiological indicators to validate some of the results that we're getting. And so just briefly, you've heard a lot about it already. We use normative modeling. Our definition of a healthy cohort, like I said, the UK Biobank doesn't necessarily have super clear labels of healthy versus patients. But we defined our healthy cohort as a minimum score on the recent depressive symptom scale, no history of depression, so 0 previous episodes, and no current medication. And these are all based on self-reported information. I'll come to the definition of the cohort in some of my next slides, and there are covariates included in the model: rate, sex, site, and head motion. And before I delve deeper, this is work from a really talented graduate student in my lab called Kayla Hannon. She's here this week, and she is presenting her poster, and she's also got an oral presentation. So I'd encourage you, if you're interested and you want to delve a little bit deeper and talk to her, I'd encourage you to seek her out. So when we were thinking about heterogeneity in depression, we think that heterogeneity might arise from different sources, and one of those sources might be clinical heterogeneity, which is what we often think about. So these are two kind of hypothetical patients, patient A and patient B, who have the same diagnosis of major depressive disorder, and their symptoms are listed here, and instead of the game of find the differences, here's the game of find the similarities. And if you stared at this for a little bit, you might see that they have one symptom in common, which is a pretty vague symptom. And this is a made-up example, but it's not unrealistic given the clinical diagnostic criteria for major depressive disorder. But another type of heterogeneity might be this example, in which we're comparing patient A to patient C. And now in this case, they have exactly the same diagnosis, and they also have exactly the same pattern of symptoms. And but yet, despite that similarity in the clinical presentation, these patients might still differ from each other in terms of their brain-based changes, their biological basis of their depression. It's a simplified example here. We all know that depression is most likely much more multivariate, and it's more of a pattern of brain changes that contributes to the symptoms. But the idea is the same, that even though they might present clinically in the same way, there might be different biological mechanisms that are contributing to that. And there might be some mapping from clinical heterogeneity onto biological heterogeneity, but there might be some residual information there as well. And so that's how we approach this project, thinking, well, maybe when we're trying to address heterogeneity in depression, maybe we should first of all try and identify groups that are clinically homogeneous, but then still test within those groups whether there's any residual biological heterogeneity. And so those were some of the questions that we wanted to ask. And another question that you might have is at each of these steps, particularly at the clinically homogeneous steps, it might be that people are already sort of have larger effect sizes in terms of their brain bases. And so instead of identifying people who have similar or the same patterns, complex patterns of symptomatology in depression, we wanted to isolate different symptoms because it keeps it a little bit simpler, and also because it gave us the opportunity to look at whether different symptoms were associated with different brain correlates. And so we looked at 3 symptoms here: anhedonia, low motivation, low mood, and somatic disturbance. Now, in the majority of patients, these symptoms are highly correlated, and the majority of patients with depression suffer from a combination of these things. But when you've got a population sample of thousands of people or tens of thousands of people, you can find those individuals that are score highly on one symptom, but low on all of the other symptoms, and that's what we're doing here. We also looked at some other clinical features of depression, for example, chronicity versus acute severity, where chronicity is the number of lifetime episodes someone has, and acute severity is their depression score right then on the day of the scanning. Again, usually those are highly correlated. Those who are more likely to be depressed on the day of scanning had more episodes. But again, we can split those out and look for people who happen to have had a lot of episodes but not be depressed on the day of scanning. And then the line at the bottom here is the heterogeneous groups, and that's actually the most common group of patients who meet our criteria, having had some episodes of depression and having an elevated score of current depression, but who have some mixture of symptoms and all of these other things. And so now the first question that we wanted to ask is, do these clinically homogeneous group differ systematically from the heterogeneous group, patient group, in terms of their normative deviations? For example, are the sample sizes larger when you correct for or control for some of this clinical heterogeneity? And also, might there be differences in the patterns of brain bases across these different clinical groups? And so I'll show you some results here. On the y-axis are the normative deviations. So these are the results from the normative modeling compared to the healthy control group. We fitted that to 80 different multimodal imaging measures, and you can see some of the results here. So the last group here, the last bar in dark red, is the heterogeneous group, and you can see that all of the clinically homogeneous groups have larger effect sizes than the heterogeneous group at the end. And you can also see that there's some different patterns across some of these different clinical groups. And then I'll expand and show you all of the different imaging measures that were significant after multiple comparison correction. And you can see these results are the same across all of these imaging measures, where the heterogeneous bar, the one at the end, is smaller compared to most or all of the clinically homogeneous groups. And you can see really interesting patterns start to emerge in terms of different clinical features mapping on to normative deviations in different brain measures. And then the last thing I'll talk about, so the next thing we wanted to do is to look, once we have these clinically homogeneous group, is there evidence for residual biological heterogeneity? Are there maybe still biological subtypes within these quite well-defined, well-characterized clinical groups? And like I said, this is ongoing work, but I'll show you results for one of the groups to give you a little bit of a teaser. So this is the acute severity group, which means that these people were high in their depression score on the day of scanning, but low in some of these other measures. I'll not discuss all of the details of the pipeline. Again, I'll encourage you to attend Kayla's talk, but she did a lot of work to identify the best number of biotypes, first of all, using bootstrapping, and then she did a lot of work in terms of cross-validation to ask the question, how stable are these biotypes depending on the data? And so the adjusted RAND index gives you an indication of stability here. But then, of course, the harder question is, are these biotypes clinically meaningful? And so the last thing that we're still actively working on and that we did here was look at 5 different measures, summary measures, that we believe to be relevant because they are related to some etiological processes that might lead to depression, to test whether the biotypes differed on that. And so the results here are interesting. These 2 biotypes differed on this summary measure of cognition, which is promising given that depression, particularly in a late life sample, has been linked to higher risk for Alzheimer's disease or potentially a prodromal symptom of Alzheimer's disease. And so with that, I'll summarize to say that individual differences in mental health come from a lot of sources, and with data sets like the UK Biobank, we have a really exciting opportunity to study that in all of its messiness. And I like normative modeling because it gives us the opportunity to standardize look at some standardized measures and offer a lot of sensitivity in terms of looking at differences from the normative population. And yeah, we're still doing ongoing work in terms of looking into these subtypes and identifying and validating their clinical value. And so with that, I'll say thanks to the collaborators on this project, to my funding and my lab, and in particular to Kayla Hannon, who is leading this project, and again encourage you to attend her oral session tomorrow in the Disorders of the Mature Nervous System session. Thank you.
Thanks very much, Janine. The next speaker today is Johanna Beyer, who's a PhD student at Origin, that's at the University of Melbourne in Australia, and she's going to talk a little bit about some site variations in normative models.
Yeah, thank you, Andre. So I'm going to shift gear a little bit with my talk, as Andre said, and I'm going to show you how you can use normative modeling for side effect correction. And this is part of my PhD project. So the background for this project is that there's a trend toward collecting large neuroimaging data sets by pooling data across studies. However, data sets that have been created in such a way often contain side effects or batch effects. And unfortunately, these effects can be correlated with biological variation or other variation of interest, for example, age, sex, and site. And because this problem is quite known, statistical methods have been developed to harmonize for these effects. The most prominent of those is the combat approach. However, these methods cannot accommodate for the correlation between side effects and variation of interest, which has been shown to lead to overconfident results in downstream analysis. I've plotted here some side effects from the Abai dataset. So the Abai dataset contains, I took Friedhofer data of 35 ROIs, and each of these box plots here is basically one individual and these 35 ROIs. And what I did is I colored them by site, and I grouped them within site by age. And you see, so this is structural cortical thickness, you see some decrease in cortical thickness over, like with age, but you also see these shifts between sites. And that's the side effects that I'm talking about. We have already heard that normative modeling aims to map the full variation in a cohort, allowing to express each individual's score as a percentile or z-score of deviation from the norm based on a set of predictors of choice. Normative modeling, if you set it up correctly, for example, based on hierarchical Bayesian models, also allows for the separation of different sources of variance. And these two facts are quite important for what I'm planning to show you. So if you include site as a predictor in a normative model, this allows you to calculate z-scores that are normalized for site, basically, because the z-scores are normalized for your predictors, and the z-scores that you get if you include site as a predictor are normalized for site. And I've just put here the most basic equations for this kind of method here. So basically what you do is kind of you train your model to make this prediction plus some noise, and then you have here a matrix with your variation of interest, for example, age, sex, disease status, and then you add another matrix for site, and then in my case I added some nonlinear component gamma. And then what you do is basically to calculate an individual z-score for an individual is this individualized prediction from your model minus the true value divided by sources of variance. So in this case it was meaningful variation and then epistemic, so noise. And that gives you the z-score. So much to the theory. Now I wanted to test whether this actually works. In order to do this, I took an open data set, like I already said, the Abide data set, and I use cortical thickness data from 35 RIs from this data set. The Abide data set contains data of over 1,000 individuals, and we have 569 healthy controls and 482 individuals with autism. And most importantly, the Abide dataset is a multi-site dataset, so it contains data from over 20 sites. And what I did to test this idea that I can use normative modeling for side effect correction is I took a rigorous train test approach. So I took the Abide dataset and divided it into the healthy control arm and the autism arm. And then I divided the healthy control arm into a training set, which was 70% of the data, and a test set. And then I have as another test set the autism arm. And then I trained my model on the training set and made predictions for most of the test sets. So now I developed two different types of normative models. They're both based on hierarchical Bayesian modeling. But one of them is basically a linear model, so only allows linear associations between the model and cortical thickness, and the other one contains a Gaussian process model, so allowing for non-linear associations. Then I thought I would like to know whether my normative modeling approach actually compares to other methods that are out there to correct for side effects. And I looked at different methods that I could find, like many people use a residualization approach, so just regressing outside, like spoiler, I would not recommend that. And then there are combat. Combat has the option to preserve age and sex. And then there's a new version of combat, combat GAM, which is a nonlinear version of combat. So the problem, if I want to compare these two types of models, like the normative models and the comparison models, is that from the normative models I get z-scores, and from the comparison models I get a corrected score in the raw scale, basically cortical thickness. So I have to do basically a second step with these type of data and pipe it through another normative model that just converts the data into z-scores. And then I have z-scores on both sides and can compare them. I looked at several measures of model performance to compare the results that I get from these types of models. There are the Pearson correlation coefficient, the standardized root mean squared error, the log likelihood, and the explained variance. And these four types of comparison methods basically look at the difference between the predicted mean and the actual value. However, I get variance scores for my models. And here I look at the mean standardized log loss and the predicted variance to look at those. And to summarize, my models outperformed all other comparison models in types of these like linear comparison methods. And then the more important one or more interesting one are the variance comparison methods. So the mean standardized log loss is basically tells you whether your prediction that you get from your model is predicts the data better than the mean of your training set. And lower values are better, and like especially values below 0 are better. And these are histograms of the 35 RIs, and you see from my two Bayesian models, almost all RIs are below 0. Whereas especially for the combat without covariance and for the residuals model, you see this is like not good. And there we had like scores that were like somewhere here. These are kind of type of okay for some regions, but yeah, these clearly win. Another interesting result that we found is if you just plot the predicted values that you get from the models against the true values here on the y-axis, what you see is that for my hierarchical Bayesian models, the dots lie nicely on one line, whereas for all the other measures, you find that there's quite a shrinkage in variance. basically. So the models take out a lot of meaningful variance. That's how we interpret that. Yeah, then another question is like, can we still predict side effects after correction with my hierarchical Bayesian models? And I've just here plotted box plots. So you see here on the x-axis, these are the 20 sites. This is like the raw data. You see this is both for the healthy control alternate test set, quite heterogeneous, as well as for the autism test set. And then after correction with my models, you see, of course, for the training set, it's awesome. And interestingly, for the autism set, it's also quite nice. For the healthy control test set, there's still some heterogeneity, but it's definitely reduced. And that's probably due to the train-test split kind of differences. Yeah, to summarize, common side effect correction methods led to an undesirable shrinkage of variance due to the removal of variation of interest. Including side as a predictor, however, in a normative model is an alternative method to common side effect correction methods. And this method also showed valid results in a clinical sample. For this paper, I used a Gaussian process to allow for nonlinear association between predictors and cortical thickness. However, Gaussian processes are known to be quite computationally costly. So for another paper, I just implemented a spline version of the model that runs significantly faster. And if you want to check all this out, what I've just shown you, which was actually just a small part of the paper. So this is the paper that, basically the stuff that I've showed you. This paper is quite opinionated and says normative modeling is great, because normative modeling is great. For side effect correction methods, this one here tries to give a bit of a more balanced perspective over side effect correction methods. Yeah, and then I thank you for your eye up. So yeah, of course I have to thank my supervisor, Liana Schmal, who is my primary supervisor in Melbourne and sadly cannot be here. Andre Marquand, who is the project supervisor for this project, and Richard Dinger, who has also contributed majorly to this project. Yeah, thank you for your attention.
Thank you. And last but certainly not least, I'm very pleased to have Mohammed Habas in Symposium with us today. He is an assistant professor at the University of San Antonio, Texas. He's done some really great work on using these kind of models in aging and dementia, and I'm very pleased to have him with us today.
Yes, thank you, Andre, for the introduction and for taking the lead in organizing this very nice session and putting this nice team together. I'm really excited to see so crowded hall here today at this late hour. My name is Mohammed Habas. I'm assistant professor of radiology and epidemiology at the University of Texas Health San Antonio, where I'm leading a neuroimage analytics lab. I'm also co-leading the neuroimaging core of our South Texas Alzheimer's Disease Research Center with Peter Fox. And I'm keeping affiliation with the University of Pennsylvania, working closely with my previous mentor, Professor Christos Devatsikos, who is leading the iStaging Consortium, and most of the results that I'm going to bring today are from that consortium. I will be talking about the brain aging charts, neuroimaging, and machine learning define normative trajectories. The way we are charting brains is to build preclinical signatures for age-related diseases using machine learning models. And I'm going to speak about 3 examples that we have derived from the iStaging Consortium today. The first example is a signature of brain aging, the SPER-BA index, which is predicted brain age. There was yesterday a whole symposium on that topic in which the principle has been explained, and it is really simple. From an MR scan, we are predicting individuals' age, and those who are looking older on MRI scan are actually advanced brain agers versus those who look younger on MR scans. We call them resilient to brain aging. Other indices that we are calculating the spare AD index, which is quantifying AD-like atrophy in the brain using machine learning and classification models. And the third index that I will be talking about is total white matter hyperintensity's volume, which can be seen as a proxy for vascular brain injury. So charting the brain might require a wide range study, and this is a report which we have put a couple of years ago in Brain with the Christos, in which we have studied the spatial distribution of white matter hyperintensities in the study of health in Pomerania, with wide age range from 40s to 90 years old. And there we have seen that white matter hyperintensities start to appear in the 40s. In frontal periventricular areas, and with age, the probability of having lesions would increase. We have studied there the risk factors that would be associated with the appearance of white matter hyperintensities, and at that time it was known that those lesions would be associated with the gray matter atrophy. But we took an approach, an innovative approach actually, to summarize brain age acceleration with white matter hyperintensity. So we looked at the association of those lesions with brain aging, as well as potential association with the spare AD index. What we found was exciting. Those who have high vascular lesion load have significantly more brain aging patterns at older age, after the age of 65 years old. And even before that, this relationship was stronger at older age. We have seen also that those who have high vascular lesion load would have more AD-like atrophy, as demonstrated in the figure below. They have higher spare AD values, which is indicating more AD-like atrophy. Well, this was a single study which had a wide age range, which is great. However, it still has limitations. Since we are not able, of course, using the same study to replicate the findings, we had to look into other cohorts towards that. There is a nice paper from Thor Wager group published in Nature Neuroscience that is summarizing and describing the need for generalization of machine learning models. So that was one thing that we were not able to consider using SHIP alone. Another problem, particularly for Alzheimer's disease and dementia, SHIP lacked in hallmarks of Alzheimer's disease, like amyloid and tau pathology. It was not, or cognitive testing, sufficient cognitive testing was not there. So what we have done later in the iStaging consortium is to plan pooling data together and access more cohorts to first generalize findings, build more robust machine learning models, and second, complete the picture and understand how brain age and brain aging would be associated with dementia. So here we have put more than 10 cohorts together and with more than 8,000 MRI scans. We have used harmonized processing pipeline. We have used afterwards a harmonization technique for the processed data, Combat GAM, which has been mentioned recently. And after harmonization, we have built brain age model across all these cohorts. And this figure here is showing a brain age for healthy individuals. We can see that advanced brain aging would be associated with extensive widespread atrophy. And risk factors is something that we are also looking at. It looks like diabetes would add 3 years of brain age, for example. We can see that from this figure. The other index that we have calculated was spare AD. Again, more positive spare AD values would mean more AD-like atrophy, and negative spare AD values would mean more normal-like brain. This figure is showing that in midlife, high spare AD would be more heterogeneous in individuals who are younger. And the patterns that are actually captured with the spare AD index are typical AD atrophy in medial temporal and parietal regions. Then we have quantified white matter hyperintensities using a recently developed deep learning model, which we have trained with data from 15 different scanners. And as we see here, I mean, this is an example where one study would not have wide age range. I think the green dots here are the UK Biobank, but can be covered at the age range from 20 to 40 by another study like the SHIP in this example. Now, after deriving all these three indices in the iStaging consortium, we came up with charting plots that we are deriving just like usual heat maps. So it is a heat map that is showing an association between two dimensions and a third dimension. In this figure here is executive function. Our brain charts are on the x-axis age. In this example, on the y-axis is predicted brain age, the SPARE BA index. And the third dimension here that we are actually plotting in the heat maps is executive function. We also plot isocontours that are fixed values for the third dimension, and the direction of those isocontours would define association with the x or y dimension. So these are the examples here in this figure. 1 and 2 would show either association with y or x. And if the isocontours have a diagonal direction, that would mean association with both dimensions. So this figure, for example, is showing that executive function is actually associated with both, with age and brain age. And at 13 years, younger brain can maintain same levels of executive function. All right. Now, can we replicate what we have seen in the SHIP study in the larger consortium? Do white matter hyperintensities associate with accelerated brain aging and with AD-like atrophy? And this is what we are plotting now precisely using the brain charts. The figure to the left is showing the association between white matter hyperintensities on the y-axis, and the third dimension here is brain age. So for an individual who is 61 years old, having more white matter hyperintensities could add up to 10 years of a brain age on average. Maybe normal values for that individual would be 10 cubic centimeters, so then the brain age would match the actual age. But the moment that we add more white matter hyperintensities for this person, we will have higher brain age values up to 70 years. The direction also of these isocontours demonstrate association between white matter hyperintensities and brain aging. On the right chart, we see relationship between the white matter hyperintensities and AD-like atrophy. So yeah, we were able to replicate those findings. But now we are also able to complete the picture, and we are also able to see association of white matter hyperintensities with cognitive testing. Again, that was not possible in the SHIP study alone. But in the iStaging consortium, we were able to find at least 2,400 MR scans with executive function. And here we see that white matter hyperintensities is indeed associated with executive function as well as with memory. And the message here is low white matter hyperintensities could maintain cognitive performance for maybe 10 years. One very exciting finding that I'm bringing up today is that having white matter hyperintensities would double the risk of testing A-beta positive. So we have taken people who have A-beta positivity either with PET, measured with PET or CSF from iStaging, and this data is from healthy individuals. Someone who is 68 years old would have the probability of having A-beta positivity up to 20%. If we add white matter hyperintensities to that brain, then we will get up to 40% A-beta positivity probability of testing A beta positive. So yeah, I mean, that was a very strong relationship. Now, what are the next steps? We are bringing those dimensions and measures to other consortia, and these are data that we are submitting soon from the AD consortium with more than 400 participants on type 1 diabetes. We have derived the SPER-AD, the SPER-BA indices after harmonization with the iStaging consortium, and we have really a very striking finding here. It looks like type 1 diabetes is not associated with AD like atrophy, but it is associated with accelerated brain aging. And this relationship might be happening before the year of 45 years old, before the age range of this population. So the paper will be really exciting looking into that. Why is that happening? Time and explain more on that. Other consortium, we are also bringing that those methods and dimensions to is the CHARGE consortium, which is looking into vascular brain aging. And this is a recent U24 effort with Tim Homan, Paul Thompson, and Christos to harmonize the CHARGE with ADSP, ADNI, the ADRC-NAC, as well as iStaging together using these methods. And also another consortium, the Diverse VCID, which is bringing white matter hyperintensities, standardized white matter hyperintensities measures to the ADRC network. And last but not least, a recent project that I'm excited to talk about in collaboration with Ilya Nasrallah and Christos. My lab is now the reading center for the RECOVER consortium, which is looking into the impact of COVID-19 on the brain across 30 sites in the US. So in summary, large N would help machine learning models to be robust, and white matter hyperintensities seem to be associated with accelerated brain aging. We have seen that in a single cohort. We have seen that in 10 cohorts, and we keep seeing that. AD-like atrophy as well is associated with white matter hyperintensities. Not only that, but those lesions were associated with lower cognitive performance. They could double the probability of testing A beta positive, and overall machine learning and neuroimaging could help in precision diagnostics. With that, I would like to thank the champion of the iStaging consortium, Ray, who is now a PhD student. I would like also to thank the leader of the iStaging consortium, Christos. I think he must be here somewhere. All collaborators, funding agencies, all cohorts and cohort participants. And I would like also to thank you for sticking with us till this time. Thank you.
Go ahead.
All right, thank you. I really enjoyed that set of talks. I hope everyone did as well. It's nice to see everyone here. So we've got some questions on the app. Feel free to use those questions if you want to. We'll be looking at that. We're also happy to have people come up to the microphones if you'd like. But I will start us off with the most popular question, which was about defining the normative population. So the question is, how do we form a representative normative population reference? What are the criteria? What should we be thinking about in terms of sample size, well-being, anything else? You want to jump in?
Yes, this is really nice question that, I mean, since we are trying to study or to chart the brain, as Andre has shown with the growth charts, I would say that representative cohorts with age range spanning the adulthood lifespan or even before that would be something that we should be looking at. Particular particularly for our case, where we are building brain aging models, we are deriving brain age models from healthy individuals. Now, the question is, what is healthy? This is something that can be debated, since some diseases like hypertension might be very prevalent after the age of 50 years old. Maybe 50% of the population would have lesions. Even amyloid positivity, I mean, we have seen from our data that maybe 20% is the norm to have A-beta positivity in people who are older than 65 years old, and this probability would increase with having other comorbid conditions. So the question is, what is healthy? That's a very difficult question, and I tend to answer that with lacking of clinical symptoms in the case of dementia. I mean, we are building our brain age models from those who do not have dementia symptoms. In the case of the SPARE-AD index, we are deriving the index from those who have AD, clinical AD diagnostics, as well as A-beta positivity. That's considering the definition of Alzheimer's disease according to the NIA guidelines at the moment. We are also taking the controls in that model to be A-beta negative to make sure that they are not contributing to some noise in the signal. But yeah, I mean, maybe it would vary according to the index that we are going to derive.
And just to add to that, I mean, you're free to, as a researcher, you're free to choose. So you can have a reference that's defined from an at-risk cohort. It's in some sense also a question of data availability. You know, if you bear in mind with the inferences that you define afterwards, that this was defined with a cohort with a certain prevalence, etc., as long as you balance your inferences with regard to that prior information, then it's fine.
Yeah, and I would like to add that basically normative modeling allows also very nicely for this dimensional approach, basically where we say that we don't only have like the categories healthy and not healthy, but maybe like we take as a normative reference population like basically a spectrum from healthy to non-healthy, and maybe like less healthy individuals then fall more into the extreme percentiles of the spectrum. So we had actually reviewers asking that. I think Andre has been asked to add basically a population where we have like also or like individuals that were more on the unhealthy like side, basically.
Yeah, we've actually done that a little bit in our project that we were working on, defining the normative population in different ways. And indeed, if you include more people on the unhealthy side, I mean, it affects some aspects as you would predict in terms of if you include symptoms, then the symptomatology score goes down a little bit, but it doesn't seem to change the fundamental results in a drastic way.
Are there any live questions? I mean, if anyone wants to just jump to the mic and ask, please feel free to do that. Otherwise, I'm going to ask you, Andre. Uncertainty estimation is known to be problematic. How did you validate your uncertainty estimates?
That's another great question, and I would answer, yes, it is. It is difficult. Typically, I mean, what you want to do is you want to separate modeling uncertainty from variation. So the difference between the two is that the modeling uncertainty should get smaller as you add more data, and that, you know, measures stochastic things in your modeling. Variation is variability across individuals, and that's what you actually want to measure, and you want to minimize the former and maximize it, or accurately quantify the latter. I would say how to validate that is an ongoing question, and I would invite whoever asked that question to visit Barbara at her poster. So one of the things that we've done in that direction is looking at it with respect to things like test-retest reliability. I mean, testing it on independent data is always going to be a pretty good way to assess that. We tend to, in the models that we work with, we tend to estimate uncertainty in a Bayesian framework, which allows us to separate it into different variance components. It does need to be done with care. So, for example, in Gamel's framework, which is another popular technique, that's a penalized regression model, and strictly speaking, using something like bootstrapping won't give you the kind of uncertainty estimates that you need. So it's an ongoing, I would say the validation is indeed an ongoing challenge. And we do the best we can with the data we have right now, but I think there is still some work to be done in that space in the coming years.
Okay, I'll move on to another question, which is about the longitudinal results that I think mainly Andre presented on, asking most normative models use cross-sectional data to define the normative deviation. And so does this pose an issue for comparing the model to within subject change? And I guess a related question is, do you specifically model that it's, do you have a sort of within subject way of estimating the deviation?
Well, you could do that. I mean, something that most people don't know, actually, is that the World Health Organization growth charts are actually also estimated using cross-sectional data, mostly. There's a little bit of longitudinal data in there, but most of it's cross-sectional. I mean, ideally, having lots of longitudinal data is really good, but to do that in a developmental setting, there's practical limitations, obviously. I mean, you don't want to wait 90 years for someone to go through a whole lifespan. If the sample size is large enough, you can get estimates that I would say are good enough, and we can also validate that in terms of again looking at against things like test-retest reliability, where you expect that there's no developmental changes across a short age range. Remind me of your question again, Janine, the related question.
Whether you can build the model specifically so that it knows that it's the same subject, so that you can take what kind of fixed and random effects into account.
Yeah, certainly you can do that. So taking it and integrating it in a random effects modeling framework would be one way to deal with that. So mixing longitudinal data and cross-sectional data would be a good way forward as well.
I think we might have a question in the audience.
Yeah, we have a live question, so please, sir, go ahead.
Yeah, sorry, it's quite loud. This question has to do with brain age prediction. So when you do brain age prediction on healthy brains, it's a supervised problem. But in a sense, when you do it on someone who has like a neurodegenerative disorder or some other problem with their brain, it becomes unsupervised in the sense that there isn't like a true exact mapping from brain to brain age. So I was wondering if, as a solution to this, you've been experimenting with like getting an average from several different brain age predictions, or if you feel like it's fine to just have like a neighborhood of where the brain age is predicted in order to deal with that.
That's a nice question. Indeed, I mean, people with dementia seems to have also accelerated brain aging. Again, I mean, when we are building the brain age model, we are keeping those dementia people outside the training sample. They are just test samples. And if you read the paper which we have just published, I did not show all the results, but actually we have also shown charts for individuals who have MCI and Alzheimer's disease, and indeed they have more brain aging.
Yeah, so the next question is... Sorry, okay.
Go ahead.
Yeah.
Great session, I enjoyed it. So I have a question for all of you, both the brain age and the normative modeling. We actually got a similar question yesterday at the brain age session. So these are great indices, but they're not specific measures. Like brain age is not specific. It could be triggered by a bunch of things, or it could not be triggered by other things that don't overlap with the model. Normative model is great, again, but something being an outlier, we don't know why it's an outlier. So what are your thoughts about this, and what is the niche of these types of indices?
A great question, and you're right that it's I mean, at the moment, the way we are doing that is in terms of post-hoc analyses. So if you can show validation in terms of things like genetics, you know, if you show that the index that you have has an increase in heritability, or you know, or that you have meaningful genetic associations with the, whatever it is that you're detecting. There is the possibility, since we're free to choose the covariates and things that we put into the model, we're free to try to increase the specificity in that way, or fit downstream models. But it's indeed a challenge. So I would see it as more in this exploratory space that you would then use as hypothesis generation for more targeted downstream models, for example, with classifiers.
I could add to that here that, again, I mean, genetics would be something exciting to extend and validate with. I mean, the spatiality index, we have published that it was associated with polygenic risk score in the SHIP study. But regarding brain age, I agree with you that that might be a limitation. I mean, brain age could be a general health index. Exciting that the type 1 diabetes was not associated with AD-like atrophy, but more with accelerated brain aging. Now, if another disease or phenotype might cause atrophy, but not in age-related regions, then I agree with you, that would not be captured with brain age.
And I think perhaps the work that I presented is an example of using it essentially as a metric, as an input to follow-up studies. And so we used it as a way of normalizing the imaging-derived measures and a way of being able to do a comparison across healthy controls, but then really delve into the patients in a lot more detail. So that's a kind of more hypothesis-driven or applications-based approach.
Okay. I'm going to ask another question from the app, and this is really the $64,000 question, and it's directed at Janine. So do you think that homogeneous subgroups actually exist, given the common assumption for most symptoms to be continuous?
Yeah, that's a good question, and the background of the project that I presented, we started off with the desire to answer the question of is heterogeneity best represented as a continuous distribution or as subgroups, and we quickly realized that it is almost an unanswerable question, and it's probably a little bit of both. So my take on it is that probably things are not clear-cut and really split. I mean, if there were really obvious subgroups, the field would have found them a long time ago, right? So I think things are more mixed than that, but that's not to say that subgroups couldn't be helpful. So I think from a clinical perspective, if you tell our clinical collaborators, oh, well, everything is just one continuous distribution, then they'll ask you, okay, so what's your recommendation? How do I use your biomarker? And so thinking about that, I believe that there are meaningful, clinically useful ways of divvying up our data that might be informative in terms of, clearly there are some patients that have more similar clinical patterns than other patients, and maybe, and potentially the same might be the case in terms of biological mechanisms, that there are some patients that are more similar than others. And if there is some way of grouping that within what is likely to be more continuous, that could meaningfully inform clinical practice, then I think that's worthwhile finding out and testing.
Good answer. Are there any other questions from the audience? If not, I'll choose one more from the list. Also, a nice question. So I'll skip down a little bit because we've talked a little bit about some of the questions are overlapping. None of the speakers talked about quality control. How does it affect the analysis results in normative modeling? I'll open up.
Happy to take that. So no, quality control is really important. It's really important. In the iStaging consortium, which is larger scale study, is really difficult to go with more than 10,000 MRI scans and the quality control every single segmentation through visual inspection. We have developed ways to do statistical outliers detection using PCA and other similar methods where we look into the processing, and those QC methods that we have developed are quite great, that most of the scans that were considered as outliers were visually inspected and either they had too extensive motion problems or something like that, so that we had to deal with. They have been excluded in the end. anything else? Or go ahead.
Indeed, it's a challenge, and we've invested also considerable effort, and Sage Rutherford particularly, in some of the in checking the quality of the data that goes into the normative models, and yeah, it's something that requires a lot of attention, particularly as you get into the outer centiles, when the clinical effects are the most interesting, and also you have the same, the highest probability of having artifactual effects. And do one more? Mm-hmm. Go ahead.
Yeah, final one.
Final question.
We haven't heard much from Johanna, so I'll pick one that went for her. And so this is a specific question asking essentially a broad question of what are some of the downsides or limitations of your approach for site correction?
Yeah, that's a very good question. As I've already indicated, there are certain ways to set up the normative model that I set up that are quite computationally costly. So I ran this Gaussian process model for 500 subjects, and it took like 1.5 days on our cluster. And then I wanted to scale it to 8,000 participants, and just, yeah, it wouldn't finish. In general, I think I would say I'm quite biased, but I think it's the best way to correct for side effects, because basically you don't take out anything that you don't model, basically. So it's quite conservative, and it also allows to model different sources of variance, so your prediction gets more noisy for parts of the spectrum where you don't have enough data. So there are very nice features, actually, of normative modeling for side effect correction. Another downside might be that it's, at the moment, the implementation is still very technical, so if you want to use it, you would have to know Stan.
Okay, thank you very much. I think we're out of time. Thank you all for your participation. I enjoyed the seminar, and just like to thank everybody, yourselves and the speakers once more.